Imagine, just for a second, the primordial Earth. Long before the first animals roamed, before plants took root, and honestly probably even before the very first single-celled organisms formed, the blueprint for life was already being written in ways we are only just beginning to understand. Welcome to this explainer. Today, we aren't just looking at everyday pathogens or standard biology. We are uncovering a cinematic story about the ultimate origin of life itself. We're going to explore a biological brain twister that has baffled scientists for over a century, blurring the lines between what's alive, what's dead, and what is something else entirely.
Okay, let's dive into this. If I asked you to name the most abundant biological entity on our planet, you might immediately think of bacteria, right? Or maybe insects. But the truth is, it's viruses. They exist in the air we breathe, the deep ocean trenches, the soil under our feet, and inside every single living creature. But viewing them merely as agents of disease means we are totally missing the bigger picture. Viruses are foundational drivers of planetary ecology and evolution. They transfer genes across species, they regulate entire ecosystems, and their origins, well, they hold the absolute key to understanding the machinery of life. Yet, for a really long time, they were completely invisible to us.
So, to make sense of all this, we're heading into Section 1, the discovery of viruses, an invisible enemy.
To truly grasp how bizarre viruses are, we have to transition backward in time. Imagine we're in the sprawling tobacco plantations of the late 19th century, where this mysterious blight was just decimating crops. In 1892, a scientist named Dmitry Ivanovsky took sap from these diseased plants and ran it through his Chamberlain filter. Now, this porcelain filter had pores so unbelievably tiny that it trapped all known bacteria. But to his absolute shock, the filtered liquid still infected healthy plants. Whatever was causing the disease was impossibly small. A few years later, in 1898, Martinus Beijerinck confirmed this, noticing the agent only multiplied inside living, growing leaves. Fast forward to 1935, and Wendell Stanley actually crystallized this entity, the tobacco mosaic virus. I mean, think about that. The fact that a biological agent could be crystallized, kind of like salt, was completely mind-bending. It wasn't until 1941, thanks to the invention of the electron microscope, that humanity finally captured the very first direct photographs of a virus, giving us visual proof of this invisible enemy.
Now, Beijerinck didn't just confirm Ivanovsky's work. He fundamentally shifted the entire scientific paradigm. He described his discovery using this incredible Latin phrase, contagium vivum fluidum, which basically translates to a soluble liquid germ. He realized this wasn't just a tiny bacterium or a fungus. He coined the word virus, drawing from the Latin word for a liquid poison. He recognized right then that viruses operate by a totally different set of rules. They are completely unable to reproduce without hijacking a host cell.
Moving right along to Section 2, Hypotheses of Viral Origins, tracing the ancient family tree. Because, seriously, where did they actually come from?
So, we figured out they exist, and we figured out how to see them. But where did they come from? Right now, science has three leading models. First up is the escape hypothesis. Think of your genome as a massive, super complex computer program. Every now and then, there are mobile genetic elements, things like transposons or plasmids, that can essentially copy and paste their own code, moving around within your DNA. The escape hypothesis suggests that, deep in evolutionary history, some of these mobile fragments managed to just break free from a cell's genome altogether. They wrapped themselves in a protective protein coat, escaped cellular control, and became independent viruses roaming from host to host. In this scenario, viruses are basically rogue, runaway bits of our own ancient genetic code.
And this brilliantly illustrates the other two major competing theories. On the one side, we have the virus-first hypothesis. This flips the script entirely. Proposing that viruses didn't come from cells at all, they predated them. In this model, viruses evolved from complex RNA and protein molecules bubbling in the primordial soup, acting as self-replicating units long before the first true cells even formed. On the flip side, we have the regressive hypothesis. This theory suggests that viruses were once complex, free-living parasitic cells that preyed on larger cells. Over eons, as they relied more and more on their hosts, they slowly lost their essential genes, essentially devolving into the stripped-down, hyper-efficient viruses we see today.
Up next is Section 3, Giant Viruses and Virophages, Bending the Rules of Life.
If you thought those origin theories neatly wrapped up the mystery, brace yourself, because we are fast-forwarding to some modern mind-bending discoveries. 30,000, that is how many years a virus called alpha P thovirus lay completely dormant, frozen deep in the Siberian permafrost, until scientists discovered and resurrected it back in 2014. And guys, this is not your average virus. It belongs to a terrifyingly fascinating category known as giant viruses. To put this in perspective, regular viruses are tiny and encode maybe a handful of genes, like the tobacco mosaic virus we just talked about only has 5. But giant viruses like alpha-P-thovirus and mimivirus are absolutely massive. They are physically larger than some bacteria, and they carry hundreds, sometimes over 1,000 genes. They even encode machinery for functions we previously thought only true cellular life possessed. The sheer scale and resilience of these ancient giants totally scramble the boundaries of what we thought viruses were capable of.
But wait, it gets even wilder. Enter the Sputnik virophage. Yes, you are reading that correctly. Sputnik is a tiny virus that actually infects a giant virus. When a giant mimivirus or mamavirus infects an amoeba, little Sputnik tags along and literally hijacks the giant virus's own replication factory. It causes the giant virus to produce abnormal deformed capsids. It basically makes the giant virus sick, decreasing its infective yield by a massive 70%. The bizarre concept of a giant virus catching a cold from a smaller virus just proves how layered, dynamic, and crazy complex this acellular world truly is.
Which brings us to Section 4, Unveiling Viral Dark Matter, the Deep Unknown. Let's shift our lens from the micro level of giant viruses to the global macro scale. Now, what's really interesting about this is the sheer exponential explosion of viral genomes added to our databases up to 2023. Look at that growth. It is a staggering explosion of new discoveries. We are finding hundreds of thousands of new viruses in the soil, in deep ocean trenches, and even right in our urban environments worldwide. This astonishing abundance of uncharacterized genetic material is what scientists now call viral dark matter. It is a hidden universe of genetic code that surrounds us constantly, yet we are only just barely beginning to map its edges.
So how exactly are we finding all this dark matter? Mainly through omics-based methods. Historically, you had to culture a virus inside a host in a lab just to study it. A really tedious process, but with techniques like metagenomics and metatranscriptomics, researchers can now bypass the petri dish entirely and sequence DNA and RNA directly from the environment. This data pipeline captures the genomes of viruses that we could never successfully grow in a lab. It highlights the incredible benefit of discovering vast, unprecedented numbers of new viruses. However, there's a major drawback. We now have millions of sequences, but absolutely no idea what they actually do.
And that leads us straight into Section 5, Strategies for the Future, Decoding the Shadows. That massive explosion of data brings us to a huge hurdle. Our pipelines are labeling roughly half of all these newly discovered viral genes as simply hypothetical proteins. Basically, science's way of saying, we have no clue. So, how do we move from discovering unknown sequences to actually utilizing them? Well, the scientific community is launching some incredible cross-disciplinary approaches to decode this dark matter. Artificial intelligence tools like AlphaFold are stepping in to predict the physical structures for over 350,000 previously unknown viral proteins. By combining this AI modeling with advanced bioinformatics, researchers can prioritize genes based on their abundance and genomic context in the environment. And look, this isn't just academic curiosity. So the crucial point is, by turning the lights on in this dark matter, we are unearthing completely novel enzymes that could revolutionize biotechnology. We're finding new ways to develop highly targeted phage therapies, literally using viruses that hunt bacteria to cure antibiotic-resistant superbug infections in humans. We are turning this genetic shadow into revolutionary biological tools.
Which brings us to our final, lingering thought. From Ivanovsky's porcelain filters to giant viruses trapped in Siberian ice, and now AI predicting the shapes of viral dark matter, we've seen that viruses are the ultimate engines of evolution. They have been trading genes and shaping the history of life on this planet for billions of years. Knowing that pieces of ancient viruses are permanently embedded in our own human genome, it leaves us with a really profound question. How much of our own biology, our own evolution, and our own future might secretly be driven by this ancient viral dark matter? The blurred lines between human cells and viral evolution suggest that they aren't just some invisible enemy out there. They are, in part, us. Thank you so much for joining me on this explainer, and keep questioning the unseen world around you.